will start admitting people also.
Okay. Do you actually edit the video file in any way after the talk?
Say it again, Ben.
Gilad, do you edit the video file in any way after the talk?
Oh, no, not at all.
Not at all. Okay.
Never. I just let it be as it is on YouTube, and that's it. I just add the nice slide that Hagen prepares, and that's it.
Okay. Do you actually edit the video file in any way after the talk?
Uh-oh.
Say it again. What's the funny sound?
I think that's Gilad.
Ah. I guess we'll wait a few more minutes while people are still streaming in.
Ja, dann nutze ich mal die Gelegenheit und frage mal Matthias, wie es läuft. Arbeitet ihr wieder voll? Alles wieder offen?
Eigentlich schon, ja, ziemlich.
Und bei euch in Freiburg?
Ja. Nur das Stadion noch nicht.
So the number of participants is still increasing. So let's wait a few more moments until this stabilizes.
All right.
Okay, so what do you think? Shall we start?
All right.
Yeah. Very good. So welcome, everybody. It's wonderful to have so many of you here again for today's webinar on protein folding and dynamics. And our speaker today is Matthias Rief, who all of you probably know. Matthias already became famous as a PhD student, actually. So when he was in Hermann Gaup's group in Munich in 1997, he did what I think was the first AFM single molecule protein folding experiment. And I was a PhD student myself at the time. I couldn't believe it when I saw the paper. This was a whole new generation of experiments. And most people in protein folding wouldn't even have considered this to be possible. And so it really was a set of key experiments for starting a new field to establish force spectroscopy and single molecule spectroscopy in general in biology, right? And nowadays, single molecule experiments are a pretty mature technique that today's PhD students take for granted, right? And so in all these developments, ever since his very first breakthroughs, it was a very exciting time to follow Matthias's work, which was always at the forefront of the field. After his time as a PhD student in Munich, he did a postdoc with Jim Spudich, and then returned as a professor in the physics department at the Technical University in Munich, and rose through the ranks with breathtaking speed. It was a full professor within, I think, 3 years. And he kept pushing the boundaries of the technique. He moved to more and more complex molecular systems. He studied them with higher and higher temporal and spatial resolution. But there's also another thing that stands out in Matthias's work besides the innovative power, and that's the fabulous data quality and rigorous data analysis, which really sets standards in the field. And so it's no surprise that Matthias is highly decorated. He's a fellow of the Biophysical Society, a member of the Bavarian and the German National Academies. He already very early on is probably one of the youngest people ever. He won the Leibniz Prize of the German Research Foundation, which is one of the biggest distinctions in science in Germany. And so in brief, it's a true pleasure to have you here, Matthias. We're very much looking forward to hearing the latest news about the forces in chaperone-mediated protein unfolding and cell adhesion. But before we start, a few technical points. Please, everyone, keep your microphones muted to minimize our background noise. And if you have a question, please put it into the chat window. And either write down the question or simply write, I have a question or something, and then we will call you in the end and ask you to unmute your microphone and ask your question. I would also like to mention that this talk will be recorded and made available on our YouTube channel. So anyone who missed the talk live can watch it later on. But now, Matthias, thank you very much again for agreeing to join us and speak in our webinar. And the screen is yours.
Just wait a second. I probably have to stop the screen sharing and stop my presentation and rerun it again. So, all right. One more try. All right. Is it visible?
Yes. Very good.
Okay, excellent. So thank you very much, Ben, for these really nice words. So I'm really impressed by myself if I sort of listen to you. So, but now is not the time to disagree. Thank you very much. I'd also like to thank Gilad and Hagen for inviting me to this really great lecture series here. So I'm proud to be here, and I'd like to share a few experiments that are ongoing in my lab. And we use optical tweezers to study the mechanics of protein folding. And I'd like to show you two examples. One from protein folding, how the glucocorticoid receptor folds and how its folding is modulated by HSP7D. And in the second part of my talk, I'd like to address the question of the forces involved in integrin adhesion I'm going to show you some experiments along those lines. Those experiments have been done actually mainly by Mihai Budescu and also Marco Cuison in my lab. And I'd like to acknowledge the great collaboration with Reinhard Fessler from the Max Planck Institute in Martinsried and his postdoc Jonas Aretz. For the glucocorticoid receptor story, the two main players there were and are Thomas Suren and Patrick Mössmer. And I'd like to also acknowledge the collaboration with Johannes Buchner, without who this wouldn't have been possible. So what I'm talking about is this experiment that is sketched up, down here, where we suspend individual proteins between two beads using DNA tethers. So that's an assay that is around now for quite a while, and optical tweezers have been used to study many mechanical questions in biomolecules, but I'd like to concentrate on DNA mechanics, on protein mechanics today. And the system, the first system, as I said, that I'm going to talk about here is the ligand binding domain of the glucocorticoid receptor. So the structure you see here, and how we came to do this experiment, it was Johannes Buchner, who is studying chaperones and the glucocorticoid receptor for quite a while. And Johannes told us that it's very difficult to study this protein because in vitro, if you investigate this, folding is severely affected actually by aggregation. So you can't do really equilibrium unfolding and folding. So the study of the folding here is very difficult. And we thought maybe in a single molecule experiments, this is easier to do. So the reason for this aggregation is actually that the apoprotein that has not bound the drug has a strong tendency for aggregation. So and now it's about time that I'm telling you what this glucocorticoid receptor is really doing. So in our body, you certainly all know the drug cortisone, so an anti-inflammatory drug. And exactly this drug here, a variant dexamethasone, binds to this glucocorticoid receptor. And the binding actually activates this receptor, which then is a transcription factor and triggers all kind of downstream signaling involved in inflammation. So that is the bigger picture. And our question is now in the first part, how does this receptor fold? And also, if you look at the structure, what you see here is that this drug dexamethasone is completely engulfed in this protein. So you don't really see how this should be able to get into there. It's completely inside of the protein. So we also want to see what are the steps through which actually this dexamethasone binds here. So here the hormone is deeply buried inside the structure. This is something that we want to study. And also, of course, we have an interest in chaperones and how they affect this. And it's known that actually the hormone binding in vivo is strongly regulated by chaperones, HSP 70, 90, 40, what have you. And so that's these are aspects that I'm going to talk about today. So, and in order to study this, we really bind our DNA and the beads or to the beads in our trap, and then we can, in a controlled fashion, unfold and refold this protein, study the steps along the way. So this is a trace that we get when we unfold the glucocorticoid receptor in an off-bill trap. So the blue curve is a pulling trace. So here we load the protein, and you see in this blue circle here that it undergoes actually some fluctuations, some transitions here. It has a first unfolding, eventually to an overall length of 12 nanometer. And if you increase further the force, then it will completely unfold to a total length of something between 80 and 90 nanometers. And what was the first surprise actually for us was that if you now relax the force again, if you bring the two beads together, then you see that it starts to fluctuate again, and in fact very readily folds into its native structure. Why was that surprising? Because as I told you, in vitro, this protein has all kinds of difficulties in folding, but apparently as a single molecule, it folds very robustly. And so we can now repetitively study the folding and unfolding of this protein in the presence of the drug. And that's what I'm going to talk about. So this little first, so something is drawing here on my screen, I don't know. This little first transition that I was showing you here, this we can study also by keeping the force constant, and then we see this fast opening and closing of this little structure, this 12 nanometer structure that I was showing you before, and we can now use different forces to study actually the energetics as well as the kinetics of this thing. So from the lifetimes, we can measure the kinetics, and we can now make a chevron plot, can really study the folding or binding kinetics here, which would be the blue curve, as well as the unbinding curve, which would be the purple curve. And then we can extrapolate this to zero load as we do this, and then we get the opening and closing rates of this element in the protein, as well as the free energy. And it's already quite some free energy. So we're talking something like 10 kBt of free energy that's just in this little element that's opening and closing, and we call this a lid structure. And of course, the first thing that I'd like to show you is what actually is this lid structure. So where's that located? Which part of the protein opens and closes? And I'm talking about this lid structure because later on I will show you that the opening and closing of this lid structure is tightly connected actually to hormone binding and unbinding. So it's something that we find is important for this glucocorticoid receptor. Now, looking back at the structure, good candidates for such elements that can open under force are those that are at the ends of the protein. So the N and the C terminus were two candidates. In fact, if we look at the N-terminal 33 amino acid residues, those are colored in purple. This is exactly what's opening. This is the lid structure. How do we know? We know that when we insert at this point here 10 amino acids, unstructured amino acids into this loop or in this part here, then our curves change. So with this insert mutant, we all of a sudden see a length change that is not just 12 nanometers, but 3 nanometers longer. So what we know is that the opening and closing now has become longer. So it must involve this segment. So it must be the 33 N-terminal amino acid residues that constitute this lid. That's how we can locate this. And now I'd like to show you that the opening and closing of this lid is really connected to hormone binding and unbinding. So what I'm going to show you here, or what I'm showing you here, is a trace where we keep the molecule under a constant load for a longer time. So you see the time bar here, 10 seconds. So the load is something around 9-10 piconewtons of force. And on that long time scale, these lid opening and closing fluctuations are now really fast. So they are in fact transitions between the purple states and the states marked in blue. So this is really rapid millisecond time scale transition. So this lid is opening and closing all the time. That's what you see here. And all of a sudden, actually, the molecule changes its mind. It's open for a really long time and proceeds even to these further unfolded green states. So what happened here, actually, what happened is that the hormone unbound, and here it's binding back. And then all of a sudden, this lid fluctuations kick in again. The hormone unbinds again. And how can we tell? How can we tell that these states here really are the hormone unbound states, while the other combination with the fluctuating lid is the hormone bound state? That's easy to do. You just change the concentration of dexamethasone, so of that drug, in solution. And if we now increase the concentration from 6 micromolar of dexamethasone to 200 micromolar, what you really see is that the duration of those light blue phases or cyan phases really becomes a lot shorter, as you can see here and there. So this really is directly related to the time that it takes to bind now such a drug, and that's now drastically faster. That's why those times are shorter now. You can do this in a quantitative way and look at the binding rate as a function of dexamethasone concentration, and it's directly proportional as you would have expected. And then we can measure here, or we can come up with a binding rate of the dexamethasone to the lid open structure of 0.03 per second in micromolar. So far I've shown you that actually the hormone binds to this lid open state, but I'm also showing you now that it also unbinds from that lid open state. I mean, this you can do by changing the probability to populate this lid open state. So here you see a curve at a 9 piconewton. And if you now look closely in this inset also, you see that the lid is mostly closed, and just really for a millisecond or so, it populates this open state and then it falls back right away. So you have a very low probability for lid open and a very high probability for lid closed. And that means that you can now measure the probability for drug unbinding, so of a population of those cyan states, as a function of how often do you have the lid open state populated. And you can, of course, now change this by increasing the force. If you increase the force, you pull harder, so open probability, or the opening probability should increase. And that's in fact what happens here. This is now a slightly higher force. And you see when in this zoom here that the blue states are populated a lot more often than up here. And together with this, we really see that unbindings happens a lot more often, even though this is the same time bar. So you have a lot more unbinding events of this dexamethasone. So that's how you can really show that unbinding requires that lid open state here. In fact, you can also now measure the probability or the dissociation rate, how it changes with the probability of populating the lid open state. In fact, you can, here are our measurement data, and you can now extrapolate this to zero force. So because you could always wonder, how is that related to a receptor that doesn't feel any mechanical force? And if we do so, we can now estimate what would the probability be for this lid to be open at zero force, and we can calculate this to be very tiny, 10 to the minus fifth here, and then we would measure dissociation rates here that are in the range of 10 to the minus fifth or so, 10 to the minus fourth, fifth. And in fact, those rates that we measure are in very good agreement with what has been measured in bulk rates. So what I'm suggesting here is that that pathway for hormone unbinding and binding may be very similar in the absence of force to the pathway that I'm showing here and that I'm investigating under mechanical load. So those numbers really agree very well. So far, I hope I could show you that actually this lid element, these first 33 amino acids, regulate hormone binding and unbinding. So we have a lid open state where these 33 amino acids are unbound, open, unbound, and to this the drug, the cortisone or the dexamethasone in this case, binds. We have the affinities that we can measure here. And as soon as it's bound, this lid will close, and that we have an associated free energy gain of 11 kBT here. And that brings now the affinity down from, or the affinity up, if you will, here from 10 to the fifth nanomolar to 1 nanomolar. So it's a really strong affinity because this lid closing energy directly adds to the affinity here of the drug. So a really strongly bound state of the drug here that is now completely engulfed in the protein, as I shown you in this structure in the beginning. So we also wanted to know actually how does the full folding of this glucocorticoid receptor proceed. And in order to do so, we raised the force very, very slightly. So we raised it here to 9.7 or so piconewton and looked at those traces for a really long time. And then we saw that this protein goes through its slit opening, closing fluctuations, and actually transitions then as soon as it loses the hormone to the fully unfolded state, and it goes back again. These are really rapid transitions. If you zoom into this area of the unfolding, you see that there are several states populated. So there's this yellow state, there's a light green state, a dark green state, and all those states are actually folding and unfolding intermediates. And through this energy landscape, the protein always goes when it goes from the unfolded state back to the folded state and binding of the hormone here that then means that the lid opening closing fluctuations then will kick in. So we can now measure the free energies of all these intermediate states relative to the unfolded state by just measuring the Boltzmann weight actually at a certain force. And what you see here is the free energy of all these individual intermediates. So we call them intermediate 1, 2 to 3. So the yellow one would be intermediate 3, and then the light green, the dark green, we have the open state, the unbound, and then we have the open bound state and the closed state. And what we see here is that actually in the absence and in the presence of this drug dexamethasone, the only thing that changes is actually the lid closing. The whole dexamethasone binding just actually induces lid closing and opening, while the intermediate states along the folding path, they have exactly the same free energy, whether you measure them actually in the presence of the dexamethasone or not. So every energy of all these intermediates is exactly the same. It is not affected the drug does not bind actually to those states. So that's what I'm saying here. The hormone interacts only with the folded state. And that actually, despite the structure that I was showing you initially, where the hormone is really engulfed in the protein, it's not that the folding occurs around this drug as a scaffold, because then this intermediate would have to bind the drug, but instead it forms until the open unbound state, and then only the drug can slip in and this lid can close. So none of the folding intermediates really bind hormone. So I'd like to summarize this folding pathway so far. So we go from a fully unfolded state to through a number of intermediates. Eventually, we end up with the open unbound state. The drug can bind with a KD of 100 micromolar, and then a drug induces the closing of this lid, and that leads then to the fully formed drug-bound glucocorticoid receptor. So the summary of this first part is then, as a single molecule, the glucocorticoid receptor really folds robustly without any aggregation. Then I hope I could show you that these N-terminal 33 amino acids, they constitute a lid that controls hormone binding and unbinding. And what I didn't show you in the interest of time here is actually that if you look closer at these intermediate states of folding, then we could identify actually a late misfold state that occurs at very, very low loads. So at high forces, it doesn't occur. But since this is not really important for the following that I'm going to show, I didn't show this to you. But that may be then the explanation why actually this receptor may aggregate in solution, actually. So such states may then lead to the solution aggregation of this. So I'd rather, instead of doing this, I'd rather show you now some results that we have on the interaction of this glucocorticoid receptor with chaperones. And I'd like to concentrate on the interaction of this glucocorticoid receptor with HSP70, HSP40, and of course ATP here. And in the group of Dave Eggert, they have done really many studies on this system, and they have some models how the different chaperones actually interact with the glucocorticoid receptor. And so the idea is that the HSP70 somehow opens up this glucocorticoid receptor, or when it's open, it stabilizes the open conformation. Then HSP90 should at some point kick in together with the heat shock organizing protein HOP. And this then brings somehow the hormone then back into this glucocorticoid receptor. And I'd like to concentrate on this first part. What can we say about the mechanism of the HSP70 interaction with this glucocorticoid receptor? What we first studied, so what Patrick Mössmer did, he studied actually the interaction with HSP40 and the glucocorticoid receptor. So to remind you, so without any chaperones, we have now some force that we apply, something around 9 piconewton. We see lid opening and closing, but mostly the protein stays actually in folded conformations, loses the dexamethasone, binds back, but it rarely really unfolds much further. When we add HSP40, actually we find an additional state that is increasingly populated. So you see these green states. In fact, this binds actually to the lid open state and opens it a little bit further, but complete unfolding does not occur with this HSP40. So this green state that we mark here in this curve in green, this is actually an HSP40 bound state. And how can we say that this has to do with HSP40? Of course, we can do concentration dependence. Low HSP40 here induces these states relatively rarely. If we increase the concentration of HSP40, we get this state more and more populated. So apparently HSP40 binds to an early state after this lid opening, but then the system actually stays there. It does not unfold any further. So the picture changes as soon as we add to this HSP40 also HSP70 and ATP. So if we do so, then we observe that this molecule gets completely unfolded at low concentration of HSP70. Unfolding really kicks in. You see steps, the molecule goes down. And if the concentration is not too high, it can also happen that those steps go backwards. So here the time scale, 50 seconds. If you add more, so at 5 micromolar HSP70, the steps actually occur a lot faster. So you see 5 seconds time bar, and it goes down and it stays there. So it completely takes this glucocorticoid receptor apart, the HSP70 together with the HSP40. And even further increasing makes this shorter once more. So we can get a full HSP70 mediated unfolding in a few seconds, sometimes actually even 2 to 3 seconds, 1 to 3 seconds. This is a very fast and robust observation, and this occurs in steps. They are not always identical, but by and large, we are talking about 3 to 5 steps that we observe in this unfolding process, and this would relate to, or would translate into 3 to 5 HSP70 molecules that are apparently required for complete unfolding of the GR. I was showing you this in the beginning, that these steps can also go sometimes backwards. So if we have low concentrations like this 1 micromolar HSP70 here, we go down, we unfold, we go up again, and actually even the hormone can bind again. So you see here hormone binding goes down, it goes up. So we are in true equilibrium where basically the binding and folding of the glucocorticoid receptor fights actually the unfolding of the HSP70-40 system, and we are in kind of an equilibrium where actually both processes are equally strong and it goes up and down. So we can also show that actually the duration of those steps really is dependent on the concentration of HSP70. So what Patrick did here is he plotted the rate or the inverse of the duration of the steps down. So this would be a step down, the wait states before it steps down. And he found that those actually are really dependent on HSP70. So they become faster at higher HSP70 concentration, while the upward step, these are the dwell times actually before it's going up. Those actually are independent. Of course, it's not just the dwell times here before the upward steps, but it's also the probability how they occur. So we did a kinetic model so that we could extract just the upward stepping process. and we could show that actually this upward step is constant. It does not depend on HSP70 concentration, indicating that actually the upward steps are related to unbinding of HSP70. So when some HSP70 comes off, that part of the protein can refold, while actually the steps down are tightly controlled by binding and ATP hydrolysis of the HSP70 and 40. So to conclude actually this chaperone unfolding part, I'd like to address the question whether what we see here, whether that actually only occurs under force. You could now argue, of course, we are always applying a few piconewton of force here, so we're putting quite some mechanical energy in the system, are helping the HSP70 to unfold this GR. Would that also happen if we didn't apply any force? I'd like to address the question, what happens actually at zero force? Since I'm going to show you some pulling and relaxation traces here, I have summarized actually what happens when you consecutively pull an individual molecule, how those unfoldings actually look. I showed you this initially in the very first trace, but I'd just like to remind you. So when we pull at around 10 piconewtons, see this little part here, that's where the lid opens and closes. Now we have unfolding. So these high force unfoldings are states where the GR has bound actually the dexamethasone. Now it's unfolded. And if we go back, actually, this is basically folding back into the apo structure. In the next pull, dex has bound again because it has this high peak again. It goes back, folds back, and so forth. In some events, actually, you see that when you relax and pull, it has not bound actually the dex, which makes sense because at those concentration, the probability for binding in this low to short time of a fraction of a second is certainly smaller than 1. So here we didn't see any binding of the dexamethasone. So what you can clearly distinguish is whether a molecule has bound dexamethasone or whether it hasn't. That's just the thing that I'd like to show you in this trace. And now we did experiments where we tried to remove the dexamethasone in order to look in a test tube actually whether the HSP70 can also unfold the apoprotein at zero force. So here, when we dialyze out all the dex overnight, and then we pull on these molecules, and I'm only showing you of each molecule the first pull. So the first pull here is dexamethasone unbound. So that's typically the low dexamethasone unbound case. Also here, also there, the red traces is the relaxation curve, while here the dexamethasone still was bound. So every now and then you still have a dexamethasone bound, but most are lost. So that's the sample that we wanted to use. And then what we did is we added to those dexamethasone unbound molecules, we added the HSP70-40 ATP system. And if we did this, actually only after 1 hour of incubation, we saw that every single first pull, so these are again the first pulls of the molecules, are completely unfolded. So this is just entropy elasticity, unfolded polypeptide. So that shows clearly that actually this unfolding also happens in the test tube. You do not need the assistance of the force, and all the mechanisms that we suggest here are likely relevant also in the absence of force. So I'd like to summarize the second part. I hope I could show you that the HSP40 can bind actually after the hormone has unbound, and that if you then add HSP70 to this system at 40, it's an active unfold and unfold days, and stepwise takes apart the whole molecule. And what our results also suggest is that we need 3 to 5 HSP70 molecules for a full unfolding. And in the remainder of the time, I'd like to switch gears and show you some of our work where we study the integrin-mediated adhesion of cells, of course, on a single molecule level, because that's what we do. So here we see a scheme of how integrin, the integrin adhesome looks like. Of course, strongly simplified here. So you see the integrins that have two chains, alpha and a beta chain. So the lower part is inside the cell, the lower part is outside of the cell, the upper part is inside the cell, and you see that the integrin on the outside can bind to fibronectins when it's open, and then transmits the mechanical strain to the inside, where it's then connected through the protein talin to the actin cytoskeleton. And the idea is actually that integrin really is also a mechanical connection here and is able to transmit forces from the outside to the inside and vice versa, and also is involved in force signaling from the inside to the outside. What we look at, so now everything is inverted. Now the extracellular side is up here, the intracellular side is down here. What we are interested in is first adhesion actually between integrin, the tail, the cytoplasmic tail of integrin, how it binds to the protein talin. And we actually looked at just an individual domain, and I'm going to show you that the interplay between talin and kindlin really modulates the integrin adhesion strongly. So we wanted to know in the first part of the experiment, how strong is that connection? Because all these force signaling models, they tacitly assume that actually the bonds, all the chain of bonds between the outside and the inside is stable. Because if they are dynamic and weak, they would break, and there would not be anything like a stable connection between inside and outside. But many of these, or much of this information is not really known. So we wanted to just measure the simple question, or just answer the simple question: what is the force actually that the integrin tail binds with actually to the talin domain? So here's the structure of this bond, the integrin here in blue and the talin here in orange. And what we did is we made a construct that where we can now really measure directly the binding strength between the talin and the integrin. And we actually made a single chain construct where we link the integrin tail piece to the talin, and we pull at cysteines that we introduce in positions which we assume kind of mimics the force application direction as it occurs also in a cell. So we are now pulling on this, and we can directly measure what is the integrin talin binding force, and we can also let it rebind because it's still tethered. What happens actually if we do this here, a constant force or constant extension experiments here, where we actually see the purple state is the bound state, where this integrin is bound to the talin, while the green state is the unbound state. So this is now just really binding and unbinding of the integrin to the talin. And what we were surprised actually that the forces are involved are really low. So we're talking about 4 to 5 piconewton that are really required here to unbind this. And this is by no means really a very strong bond. And what is more, it's actually also very dynamic. So we see the lifetimes here are really short. So in fact, if we extrapolate those lifetimes to 0 piconewton, so how dynamic would it be if we didn't apply any force? It would come off actually 60 times per second. So this is super dynamic and is not really strong. We're talking about only a few kVt of free energy. So this bond is not really what we thought it would be. So it's not a stable mechanical bond here. So how can we understand how this works then? First, we thought maybe it has to do that we just have a single domain. So we used the full talin head. So this requires actually all 4 firm domains, even though the binding is mostly to the third one, as we had in our shorter construct. So we made this big construct, again, applied load, and we saw that the unbinding and binding that you see down here really occurs at pretty much the same forces. There's not any difference, maybe a kVt or so, but nothing really noticeable. Here you see the unfolding of those talin domains, but that's not what we're interested in. We were interested in actually, did the full length talin head actually modulate the binding strength, but it didn't. So that's not the answer. So we looked to a second molecule. So our collaborator, Reinhard Fessler, he discovered the role of kindlin in the integrin adhesion. So he, of course, said, why don't you guys try this kindlin, which also binds actually to the integrin tail. So here you see this beta integrin tail. We so far studied this interaction between the talin and this integrin tail. And the next thing would be the interaction between the kindlin and this integrin tail. And those binding sites are very, very close to each other. So not like this drawing suggests, they're not really far apart. There's maybe 1-2 amino acids actually between those binding sites. And so we asked the question, how is that bond between talin and integrin modulated if we add kindlin to this solution? So here is a structural model. So it's not a real structure. The only structure, so this is modeled from 2 distinct structures. So this, the red part is the integrin tail. Here you see the talin integrin interaction, and you see how close this other interaction is. The kindlin actually with this firm 3 domain is very close. So here you see it even better. It's almost adjacent, so they bump almost into each other when they want to bind to this integrin tail. So very close binding sites. And we asked the question, how is the presence of the kindlin now modulating the talin integrin interaction? In the absence of kindlin, that's what I showed you. We have binding and unbinding, very dynamic. So this is the 60 per second interaction that opens and closes all the time at here a force of something like 5 to 6 piconewtons. And when my student Mihai added this kindlin protein, what he found is that the lifetimes of these bound states all of a sudden became actually double exponential. So there was short times of the binding of kindlin the short times of the binding of talin and integrin that we've seen before, they become interspersed actually with much longer times. Again, we looked just at the binding of talin to integrin, but all of a sudden there seem to be states induced that last a lot longer. And if you add more kindlin, then those states actually become more and more prominent. So apparently integrin somehow reinforces that bond between talin and integrin. So this kindlin seems to do something here, and what it actually does is this kindlin, it makes this talin integrin bond force insensitive, as I write here. So how can we tell? So we can just look at those green lifetimes that we just see from this double exponential between the purple and the green ones. So this green lifetime actually is not changing when you apply higher forces. So normally you would expect if you pull harder, the lifetimes of these green states should become shorter. So like the purple ones actually do. So the purple lifetime, this is where the kindlin is absent. These purple states, they become shorter and shorter, down to milliseconds and almost not measurable anymore. But those green states, they are always the same. So there's something like 0.2 seconds in lifetime, and they are force insensitive. So we can plot this, and we plot the off rate of the green states, it's force insensitive, while these purple states, they are really very force sensitive. So this bond lets go very easily when you increase the force, while the green one doesn't care about the force. And this actually becomes even more drastic when you go to higher loads. So we wanted to apply now really high loads. So we can only do this if we do some quench experiments. So we keep the protein at a low force for a while, and then we ramp the force up suddenly to a high force until the molecules unbind, and then we go back to low force, and we jump between high and low force states. And here's an example when we really jump up to 14 piconewtons, which is 3 times the force that I've shown you before. Those green states have exactly the same lifetimes. So to summarize this, so if we zoom out now, we see that these are the 2 new measurement points. Really, this kindlin makes a completely force insensitive bond. And now the question is, how could that be? We don't have a full answer yet. We have a model. So if you look at our pulling of this integrin-talin bond, so you see here the integrin chain, if you now look at the kindlin, you might get the idea that if you now pull on this and try to separate the purple and the red, then you see that actually this blue one will probably bump into this the purple talin. It may be that kindlin actually really mechanically shuts this off from force. So when normally actually this can slide in order to unbind, the presence of this kindlin further downstream on this tail may just prevent the sliding and make this bond really force insensitive. So the transition state position, in other words, would be close to 0 for this bond. So while here, that should be the talin and integrin, this will be force dependent. The talin can slide along the integrin and unbind. In the presence of this kindlin, when you now want to separate the red and the talin, really it can't do this anymore because the kindlin will bump into this and the force runs along a different direction, and actually this whole bond would be shut off from force, and the transition state position might move to 0. So that's our current working hypothesis. What have we in support actually of this? We tried to make a mutation where we inserted right here, or in the structure right here, a few amino acids, so as little as 3 or 4 amino acids. Then talin and kindlin still bind, but this force insensitivity is completely gone. So then this bond is just force insensitive, because that would give the molecules some slack so that they can still move, and the force sensitivity would be the same as in the case of integrin-talin. But this is still underway, and I don't want to make some really definitive conclusions here at this point. But the effect is clear, and the role here for cell adhesion would be that only together with kindlin, and kindlin is known to be really an activator of integrin-mediated adhesion, that actually kindlin really together with talin in the early formation of such an adhesion, when actually the cell ratchets over the surface, tries to make this connection and then pulls, that actually this force insensitivity allows then some time for more of those complexes to form, and together it's known that these adhesions then ripen and more proteins get recruited, and then eventually this will evolve in a stable adhesion. That's our model, that this talin integrin bond, actually with this force insensitive or ideal bond, if you will, really plays an important role in establishing the adhesion in early stages. And so I'd like to summarize this. So kindlin and talin individually form mechanically weak bonds, but kindlin makes actually this bond force independent, which I hope I could show you. And this may be really important in early events of adhesion formation. The mechanism of reinforcement is still something that we need to do. And at the moment, we have also in our collaboration, Reinhard Fessler's group, they are trying in vivo testing of this model. And in fact, those mutations that I was mentioning, they have a drastic effect. So there may be really effects also that we are able to test in vivo for that, but that's a little bit further down the road. And with this, I'm done. I thank you very much for your attention, and I'm happy to take some questions.
Thank you very much, Matthias. It is really quite amazing, the amount of detail you can see, and also the new mechanisms you can discover with this enormously powerful technique. Thanks a lot.
Thank you.
So we already have a few questions here in the chat. The first one is by Stephen Fried.
Yeah, no, this was really wonderful. Thank you so much for sharing. I wanted to ask a question. It's a few slides back when you were talking about the HSP70 mediated unfolding mechanism, in particular, the portion where you were showing it refolding, sort of like slowly back up. Mm-hmm. And the question that I was wondering is, so I know that in the normal catalytic cycle, you would have like a nucleotide exchange factor, and that would help swap out ADP for ATP, and then that would sort of allow HSP70 to kind of like let go a little bit. And then that would, I would assume in the normal biological process, be the situation where the protein would then be able to refold. And I was curious if your experiments had that factor, and maybe I missed it, or if not, do you think that you're looking at the sort of the endogenous sort of nucleotide exchange rate, just happening spontaneously?
This is a very good question. Thank you for asking this. Actually, we tested this. We also have measurements where we actually added the nucleotide exchange factor of the system, and it's exactly as you're saying. So this makes those backward steps faster.
Okay.
So it really sort of goes then up and down a lot quicker, and that may be very important because so the picture that I've drawn so far may sort of convey something where it totally kills the protein so that it unfolds and hangs out there for a really long time. But if you have the nucleotide exchange factors in reasonable good concentrations, it might be way more flexible. It might come back much easier. And that's in fact what it does in the experiment. So a lot quicker up and down. Yeah, it's a very good point. Yeah.
It's cool because but it also shows, which is really neat, that like HSP70 can also spontaneously lose ADP, which I don't know if I've ever seen an experiment that's shown that before. So that's really nice.
No, it can. It has an ADP dissociation rate, but it's slow. Yeah.
Yeah. All right. Thank you.
And our next question is from Gilad.
So Matthias, as Ben said in the beginning, really wonderfully beautiful data and impressive analysis as well. So I have a question about the glucocorticoid project. You showed that the open time is dependent on the concentration of dexamethasone. But it seemed to me that the close time was also dependent on the concentration of the drug. So how can that be? Or am I wrong in interpretation?
So where did you conclude that from? So I mean, when I say about the open time, then I'm talking about the open unbound. So only when it can also open and not lose the dexamethasone. This time is independent of anything. But if it's open and then the drug comes out, these were these cyan states. Maybe I can sort of share the screen again to
the trajectory that you showed at the close time that also became longer, if I remember correctly.
So let's see whether I can. So here, this would be one of those trajectories. And so if we have more dexamethasone, then we have a shorter cyan state.
Right, but you also seem to have longer violet states. Or is it
no, they are actually not much longer because we have a state here. We have a light blue state here. We have one here, maybe here also, and there. So they are, and we might actually miss even one at this concentration. So by and large, I mean, this is not a real difference here. So do you see my point? So it's not really long cyan and long purple states. They are interrupted by very short unbound states. Yeah.
Okay. Okay. Thank you.
All right.
And our next question is by Tim Craggs.
Hey there, Matthias. Lovely work there. I was interested in how you're defining the fact of the need for the 3 to 5 HSP70s, and whether you're just kind of step counting and fitting that, or whether you've actually done some experiments where you can fluorescently label those, for example, and physically count them within the system. Maybe you could elaborate on that for me.
This is an excellent point. So we are actually currently working on additional fluorescent experiment, because seeing is believing, as we know. So it would be a lot nicer. At the moment, we are inferring from the kinetics here, how many steps do we see? But you are completely right. That's why we cannot really say, is it 3, is it 5? Because also the sequence of these steps, it's not, I don't want to say that it's exactly the same intermediates. It's not like a super precise machine in that sense. We have the impression that the first step is pretty well defined, so the first point of attack, but then it might actually come from the C terminus, it might continue the way, so there's a lot of combinations. And these experiments that you are suggesting certainly would be very attractive. We have some ideas how to do this, because you need also high concentration with the fluorescence, so we have to be in the a 10 micromolar or 5 micromolar, maybe 2 micromolar range. But we have some ideas where we hopefully can do this. But so far, the conclusions come from the number of steps that we count. This is right. Yeah. Thank you.
Great. Thank you.
Can I maybe insert a brief question here, because it's along the same lines? So do you see, or do you think there are any relations between the intermediates that you see without the chaperones and with? Are they linked in any way, or is that?
This is a very, very good point, and we have spent a lot of time to look into this question. So what we find is that these intermediates are likely different. How can we say this? Because we have the kinetics of those intermediates, and also this also relates to the model. So maybe, and you didn't ask that explicitly, but the question is, how do we think also, how does this HSP70 take those things apart? So does it just grab onto like a ratchet? Does it bind in a conformational selection mechanism, the open states that happen to that the protein happens to provide, or does it induce other states? And it seems that those states are different, because we can also measure what are the fluctuations, what are spontaneous fluctuations, how often at a given state do we see excursion into the state down, what would be the probability for binding, and we don't really see that a conformational selection mechanism would be possible. It seems as if this HSP70 really pries this open. But then again, I mean, I left out a lot of these things that are really interesting here. It also doesn't really bind a long time to the folded portions, because that would be the other idea, that it maybe binds an unfolded intermediate that then cranks it open. We have done tons of really slow, careful experiments to see whether there's a binding, because the binding to these folded intermediates would involve that they are stabilized. Otherwise, they wouldn't bind. So in some way, they should be stabilized. But there's absolutely no indication. So the best model so far is something where either that binding is transient, so that binding leads to immediate unfolding, so that we can't see a binding to the folded portion, or some mechanism that also has been suggested for the HSP70 system that is something like entropic pulling. I'm sure you're aware of these models. But then that has been suggested for pores. I mean, membrane cages unfolding. I mean, there are walls that this needs to bump into, but we don't have a wall here. This is just really in solution. So our preferred model is that probably binds transiently to some folded structure yet, and then takes it apart, but that's not yet definitive.
And
yeah?
a few years ago, when we looked at expansion of substrate proteins by HSP70, we also looked for binding sites that were identified by bioinformatics. Did you see whether you find any obvious binding sites?
We saw one for the first. We have a very good candidate, and we're in the process of testing this, maybe through mutation, whether we could make this then take longer. We saw definitely enough binding sites, HSP70 binding sites, but they are not exactly in the same spacing. I mean, these prediction program, they find so many potential binding sites that, I mean, you could probably bring this together kind of, but I'm not 100% sure how definitive the conclusions really here are. But for the first one, we have a pretty good understanding, because it always seems to be at 33 nanometers, and then we can really sort of dig in there. But then the rest may be much more fluid, actually. Yeah. Mm-hmm.
Great, thanks very much. So next question is from David Jacobson.
Thanks. So I had a more technical question. In the first part of the talk, you showed this plot of transition rate as a function of force for unfolding and refolding at non-zero force, and then you extrapolated it to get the zero force transition rates. And I was wondering why that extrapolation was curved.
That's a very good question. So maybe I share my screen once more so that so here you're talking about this
Yeah, exactly.
This curved thing. And so, I mean, in very simple, or in the zero order models, if you will, for force dependent kinetics, you have straight lines. This would be the Bell model, where we would have a linear dependence, and within the measured range, it seems linear. So you might rightly wonder, actually, why do you guys see here a curvature that's not really in the data? So if you zoom in here, it's pretty much a line. And why do we nevertheless use this extrapolation? Because we know that what we are pulling on is a peptide. And the peptide has some elasticity, and we know the behavior of this elasticity pretty well. So this is a worm-like chain. So what we put in here, and that actually happens to be relevant only at low forces, is the elasticity of a worm-like chain, which would bring the transition state down at low forces, because everything would move closer together, because the entropic chain would have a recoil there. And so the answer is we model into our, or we bring into our models actually the nature of the elasticity of an unfolded polypeptide in there.
Okay, thank you.
Does that make sense?
Yeah, so it's like your distance to the transition state is changing as a function of force.
It's changing because the whole thing is elastic. Yeah.
Okay, next question by Rafael Patrosian.
Yeah, thank you for a very interesting talk. What was the idea behind tethering integrin to talin? Why not to add it just to the buffer? Because it seemed to be small enough, probably not to aggregate. Wouldn't the tethering affect possible configurations between proteins?
It actually can. So the idea is to make this measurable, so to allow rebinding. What your suggestion would be to pull on the talin and then add the integrin in solution. Is that what you were suggesting?
To pull on the talin while the integrin is in the buffer.
Exactly. So so then the effect would be very minor, because you would just measure very indirectly the stabilization of the talin, so the folding difference in folding stability in the presence and absence. That could be done potentially, but the effect would be very mild, because you would just measure how strong is talin, because that would be your readout. But I can sort of continue even. So what we did, we used actually the tethered construct, and again used that at kind of a sensor when we added additional integrin in solution. So then it would fluctuate, and as soon as the solution integrin would bind, it's open until it unbounds. And with these constructs, we can actually show that this effect is small.
Okay.
That's probably the construct that you would think of when you design this. So we did this, but I mean, I just showed you now the information on the tethered construct. For the off rates, this is good, but on rates, of course, we can't measure, because we make a first order reaction to a zero order reaction, kind of, by tethering.
Okay.
But you can do this indirect experiment where you have this in solution and use the same construct that would then block these fluctuations.
Okay, Jose Onachik has a question.
Okay. That was, Matthias, absolutely fantastic talk. I think it's just amazing what's coming out, and it's a lot of food for thought. The question I have in a technicality about it is basically, when we do unfolding and folding of small peptides or smaller systems, we know the reaction coordinate for force unfolding is not necessarily the same reaction coordinate that for thermal unfolding. So how your result may depend on where exactly you're pulling, which directions you're pulling, particularly when you do this sort of zero force expansion. That can be a very dangerous game. So how you're sure that these things make sense and the reaction coordinates about the same, and how do you choose which directions you're pulling these proteins?
Absolutely. So, I mean, we can design this. I mean, so we can certainly answer the question, would it matter whether you pull sort of in some orthogonal or whatever different direction? So the answer is twofold. So in the kind of the integrin adhesion, I think what really matters there, because there's a system where force has a natural role. So there we try to mimic whatever would probably be happening when you pull across the membrane. So there, I think also the zero force rate wouldn't probably matter too much. because I mean, as soon as this thing is loaded, then I think the assay we're doing is relevant. So there's not so much justification needed. That's my point. But for the other thing, like the GR, you're completely right. So I mean, how can we say that that when we pull at the ends like we do here, how can we tell that this is really also what happens in the absence of load? And there, I often have to rely on indications. I mean, like for example, that I could show you that the off rate that we would get of that drug when we just assume that this pathway persists down to zero load, those off rates are the same as one measures in solution. This is just an indication. It could still be that there's a number of paths, because I'm not claiming that we can measure the kinetics by a factor of 2, but order of magnitude maybe. And so this is an indication. So that's what we do here. And for some systems, we can also use orthogonal coordinates. I mean, we can certainly design it and pull in other directions. But for the chaperone system, so I showed you the experiment that, because we directly did the experiment at zero load. For those chaperones, I think is an important question. Would they take it apart even if you don't have a force? But I think there we could show that this is happening. So we always have to validate our experiments. That's kind of the...
I think that's great, because these things basically on this particular system, like you said, if you're pulling orthogonal, you really can start to give a hint if this reaction coordinates that you see with force, without force are the same. So I'm glad you're thinking about that. The other question, I think you talk about the off rate, I think is a good idea, because also tells you if your experiments are close to equilibrium, right? That you're not pulling too fast or something like that. So I think the comparing of these rates should help you to answer the question about how fast experiments done.
Exactly, yeah. Mm-hmm.
And our next question is from Tawal Chowdhury.
Hello, sir. Thank you so much for the fantastic talk. And I feel my question has already been answered, so I will try to modify it a little. So in the other contemporary single molecule optical tweezer experiments looking to study the chaperone activity of HSP70, obviously we see that HSP70 also can stabilize the completely unfolded peptide state, but also can interact with the partially folded states and stabilize them, actually stabilize them against forced unfolding in some cases. But obviously you said that you do not see any stabilization of the partially folded states.
We really
but then also
we tried to see this with tons of experiments, but we couldn't see it. Yeah.
But that might also be because you have a zero wait time at 0 piconewton in your pulling experiments.
Not necessarily. So we're going up to whatever. We vary this. I mean, we have full control over all the times, and we.
Okay, so you do not wait. So even if you wait at 0 piconewton in between the pulling cycles, you do not see any stabilization of the partially folded states.
We have even looked at experiments done at 1 nanometers per second. So we have gone as slowly as possible, and as absolutely we couldn't find anything.
That's very interesting. I think that might somehow be actually interesting, and comparing it to these recent works could be something. Anyways,
depend on the system.
Yeah. It could be actually that HSP70 works specifically in this case because HSP40 is also involved there. While in the other cases, it was only HSP70 and its co-chaperones with ATP and ADP state. So that could be the difference. That's it, sir. You already answered my question, so I'm sorry to waste your time.
Okay, Tim had another question.
Matthias, sorry, just to be greedy. I was really interested in the GR work at the beginning and looking at the opening and closing and the effect of the binding. Remind me in the data, do you see the closed conformation in the absence of the ligand?
The closed conformation we do not see. So it's all the lowest energy conformation that we can measure under force. I cannot exclude that it has a tiny tendency there to really close, but at those forces that we can measure at, there is no closed state.
Okay, and then what was did you do the experiment with the effect of your additional 10 amino acid linker? What effect did that have actually on the closing dynamics?
So I mean, the dynamics gets kind of slower. I mean, you can see this in those traces. So the fluctuations became a little bit slower, but that has also to do with the higher force sensitivity. You know, you have a longer chain that needs to contract, so that will then dominate the kinetics that we measure. Yeah.
Right enough.
Energy is kind of the same, maybe a little lower. Yeah, because we do something to the protein also. Yeah.
Can I ask another question along the same direction? So this lid, you have this little helix, and this relatively long would look like an unstructured bit. Is that really unstructured, or is that actually folded in the structure?
It is folded because it's a real crystal structure that so
Okay.
it can be resolved in a crystal structure. Yeah.
Okay. So it wasn't. Yeah, it was not.
It was not. I termed this not really correctly. Yeah.
Okay. Good. Tom Parkins had a question. No, that was just a thank you, Matthias. Okay. I think there are no other questions. So then that brings us to the end for today. Thank you very much again, Matthias, for this.
Yeah, thank you guys. It was a pleasure.
Wonderful data and wonderful analysis, wonderful insights. And thank you all for joining. I would like to remind you that 2 weeks from now, our next lecture will be given by Susan Marcusi. We'll talk about the modulation of the energy landscape by cellular factors. Please have a look at our website for the future talks. Thanks again for joining us. Thanks, Matthias, and have a good time. Hope to see you in 2 weeks. Goodbye.
Bye-bye, everybody.